Severe symptoms of COVID-19, leading often to death, are thought to result from the patient's own acute immune response rather than from damage inflicted directly by the virus. Immense research efforts are therefore invested in figuring out how the virus manages to mount an effective invasion while throwing the immune system off course. A new study, published today in Nature, reveals a multipronged strategy that the virus employs to ensure its quick and efficient replication, while avoiding detection by the immune system. The joint labor of the research groups of Dr. Noam Stern-Ginossar at the Weizmann Institute of Science and Dr. Nir Paran and Dr. Tomer Israely of the Israel Institute for Biological, Chemical and Environmental Sciences. This study focused on understanding the molecular mechanisms at work during an infection. Our cells are normally able to recognize that they're being invaded and quickly dispatch signaling molecules, which alert the immune system of the attack. With SARS-CoV-2, it was apparent early on that something was not working quite right. Not only is the immune response delayed, enabling the virus to quickly replicate, unhindered, but once this response does occur, it's often so severe that instead of fighting the virus, it causes damage to its huma. Most of the research that has addressed this issue so far concentrated on specific viral proteins and characterized their functions. Yet not enough is known today about what is actually going on in the infected cells themselves, says Stern-Ginossar, of the Molecular Genetics Department. So we infected cells with the virus and proceeded to assess how infection affects important biochemical processes in the cell, such as gene expression and protein synthesis. When cells are infected by viruses, they start expressing a series of specific antiviral genes. Some act as first line. At this point, both the cell and the virus race to the ribosomes, the cell's protein synthesis factories, which the virus itself lacks. What ensues is a battle between the two over this precious resource. The new study has elucidated how SARS-CoV-2 gains the upper hand in this battle. It is able to quickly, in a matter of hours, take over the cell's protein-making machinery and at the same time to neutralize the cell's antiviral signaling, both internal and external, delaying and muddling the immune. The researchers showed that the virus is able to hack the cell's hardware, taking over its protein synthesis machinery, by relying on three separate, yet complementary, tactics. The first tactic the virus uses is to reduce the cell's capacity for translating genes into proteins, meaning that less proteins are synthesized overall. The second tactic is that it actively degrades the cell's messenger RNAs, mRNA, the molecules that carry instructions for making proteins from the DNA to the ribosomes, while its own mRNA transcripts remain protected. Finally, the study revealed that the virus is also able to prevent the export of mRNAs from the cell's nucleus, where they are synthesized, to the cell's main chamber, where they normally serve as the template for protein synthesis. By employing this three-way strategy, which appears to be unique to SARS-CoV-2, the virus is able to efficiently execute what we call host shutoff, where the virus takes over the cell's protein synthesis capacity, Stern-Ginossar explains. In this way, messages from important antiviral genes, which the cell rushes to produce upon infection, do not make it to the factory floor to be translated into active proteins, resulting in the delayed immune response we are seeing in the clinic. The good news is that this study was also Suche. Study authors also included Yara Finkel, Avi Gluck, Aharon Nachshon, Dr. Roni Winkler, Tal Fisher, Bat-Shiva Rosman, Dr. Oral Mizrahi and Dr. Michal Schwartz, who are all members of Dr. Noam Stern-Ginossar's group, Dr. Yoav Lubelsky and Benjamin Zuckerman from Professor Igor Ulitsky's group in the Depar. Stern-Ginossar's research is supported by Skirball Chair in New Scientists. Nell Family Center for Microbiology. American Committee for the Weizmann Institute of Science 70th Anniversary Lab. Ben B. and Joyce E. Eisenberg Foundation. Maurice and Vivian Wohl Biology Endowment. And Miel de Botton. Thanks for watching. Please subscribe my channel for more updates.